everyone's on the same page. Let me see. I think I uploaded the lab. Let me just try again and do that real quick. Okay, so that is the PDF for this lab, and I think Dr. Rhoades already showed you. So I'm using the R GUI, and I'm using the R editor to, as you can see, the console is to my left, and the editor is to my right. So what you can do is to copy and paste that PDF file into this R editor, and then you can just follow down with my instructions. So the first thing you want to do is, let me also, let me see, because there's a file that you also have to put in your working directory. Let me see. Okay, so you also would need that.
Excuse me, which file?
Yeah, I'm about to put it on the chat so you can download. It's called a subset. Yeah. Okay. Right. Okay. Can you see it now?
Yes.
All right. I just sent it to so you can just download that. Just the first small file. Okay. All right. So. All right. So let's get started. So the first thing you want to do is make sure what's it called? Let me say the file path for your folder. So for example, I have a folder on my home local computer. So that's the summer 2022. And that's where I put the script and the file, the subset file and all that. And for me to be able to access that, I need to set, which is the command used, set WD, and then you can always copy the file path into into that command. That's the way I did it. You have a double quotation, have a C / users, and then till you get to that folder that has your files. So the first thing you want to do is to run this command. So you tell the computer to set your working directory to this folder, summer 2022. I don't know what you have, but I think you should be able to do that. But let me just walk you guys through it. So once you have that done, so now you're in your working directory. Now it's time to install the packages that are required. Like I think it's been mentioned before. In R, certain packages are required to draw different plots or different, visuals. So in this case for DAGs, using what's it called, the bigPint, I think it's already been said. So what you want to do is these 3 lines to hold together. So you can right click and then you run this selection. I think I already did, but that's, so it gave me this option. So now that I've what's it called, installed the bigPint package, now it's asking me to update all, some, or none of the of this package. So I'm interested in updating all. So you can just type in A in your console, although I already have it here, so I can just run this line. And then that should update my bigPint package into the current what's it called, update available out there. So it's a big what's it called, it's a big R package, so it's gonna take a while, depending on how fast your computer is. Yes, please. Rashid, right? Okay.
Yes, so Dr. Sobitan, thank you so much for guiding us through the whole process. So my question was, so right for now, just for now, we stop at library data.table, yes?
You stop at what? Oh, I didn't get your question. If you can, you say you stop at Rashid?
Yes, sorry, my internet just cut out.
Oh,
Yes.
okay. You said wait. I didn't get your question. I didn't get the last part of the
Okay.
question.
So like for now, do we just stop at just input library data.table and then stop?
Uh, oh, are you using R Studio?
No, I'm just using like the normal regular R.
Oh, okay. So this is from here. I'm not sure if you have it. This is where you stop. So
Yes.
this three lines, yeah, this three lines together where it says install the packages, the bulk manager, and then the bulk manager install bigPint. So all three of this is what you run at the same time. So that should install the bigPint R package in your system.
Also, what does the A stand?
Yeah, so the A here, so once you've run your package, it's going to ask you or prompt you to update the package. So you have the option of updating all or some of the what's it called, the libraries under that package. But I always choose to update all. That's what the AA stands for.
Oh, okay. So I just have to check if that same option appeared for me, because I don't think like when I wrote A, it didn't exactly register the. I have to go back and check.
Okay. Yeah, like I said, that's a lot of sub packages within this. So this might take a while, not less than 2 minutes or a minute, depending on the machine. And if it's already done for you, you can just go ahead and library this individual package, the big pint, and you also have the DP layer also installed, so you can library that. Have the ggplot2, you have the plotly as well, so you can library all this for packages if it's done for you. Yeah, okay. 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 This should be done by now. All right. So still running as a background. See if it's done. And just to provide more background, so the big pint allows you to have a to kind of string different scripts together, but into what is called a pipeline. So with that, you can actually using the same data set, you can do as many um, analysis as possible or data visualization as possible. So that's one of the advantage of the big pint. So it's finally done. So all right. So what I can do now is now library the big pint. Don't take too long. Okay. All right. All right. Do the deep layer, ggplot2, plotly. Please make sure you do this stepwise so you don't want to miss a step. Once any of this is not available, it means that you don't have the tool. So it's like you're working on your car and the spanner is missing. So you need to have all the tools available for you to finish this job. So if you don't library, you're just going to run into errors. I think someone just wrote something. Yes. So yes. So yeah, you can just do Control R. So doing that, just yeah. So now it's time to install another package. It's the Edge R. Is specifically for DEGs. All right. So this also will prompt you to also update, and then I choose to update all, and then that shouldn't take as long as all right. This is almost done. All right. So all the packages within the Edge are about managers being updated. And then what I would do next is to library my Edge R and then my data.table. So the data.table is also a package that allows you to work with what's it called, a data frame, and to be able to do different manipulate, can easily manipulate the data frame within itself, like what we're about to do, which we have genes. You have the first column, which is just a gene, and then the other columns are like the control, the replicates of the control, and the replicates of the treatment. So you have about 7 rows, right? The first row is, sorry, 7 columns. The first column is the gene name, and then you have those replicates. First 3 replicates belong to the control. So imagine if you're trying to look at the effect of a drug at 0 hours, at 4 hours, and after 7 hours. And for the control, you look at the cell without the drug, and for the treatment, once you've given the patients the drug, and then you look at for the treatment, you also have the same 3 replicates. Um, the effect on the cell at 0 hours, 4 hours, and 7 hours. So that's how the whole differential gene expression of genes actually the what's it called? That's how the data frame looks like. All right. So I guess this is also bringing everything in there. So all right, I think it's almost done. Then we're to get to the file. This is taking longer than usual. Let's see. Okay. Okay. So table. And then of course, once this is done, we do the library, and then the next thing is to create an object. Um, so know R works well with objects. And then for you to be able to read a file into R, you have to put them in an object. In this case, the object created is named the refstick, and then we use the read.table function. And then you include that, which is the subset.txt, which is my file, including the extension. And then you say header equals true, true in uppercase. If it's in lowercase, you'll get an error. So R in this case, kind of pay attention to that as well. So this will read my subset the text into the refseq, and then we can now start using the refseq for our downstream analysis. Just want to carry everyone along as possible. So let's see. Ooh, okay. Goodness. All right. So now let's library that. Which is good. All right. So that's good as well. Okay. So that's good. So we have everything we need. So it's now to get into the file itself. So this is the object, the line used to create the object. So that's RNASeq. And if you just want to see what it looks like, you can just, okay, there's a question. I fell behind on the scripts. I ran into an error when trying to run the line for installing. How would I go about rectifying it? Let's see. Um, let's see. Hmm, we'll fix that in the problems. Okay. All right. So let's so if you want to view what the data set look like, you can just always type that out, and then there you see. So these, in this case, we have about 100. Yeah, it's about 100. Like you said, it has about 700 observations or rows, and then it has about 7 columns. So the first three are the replicates for the control, and the last three are the replicates for the treatment. So now you see how it looks like, and then that's it. So let me just now let's begin to pre-process the data. So this pipeline, the bigPint and the edge R, they're also very specific. So when it comes to R, for example, there's no way you can add what's it called, names together. Like if you can, it doesn't work like that. So you need to know the right data type to fit into your program. For example, the gene name, the gene, which is the first column, as a factor data type. So what we need to do is to change that into a character data type. And the one liner to do that is to use these. This is the object, RefSeq ID that I'm using just to change the data type into a character data type. So the RefSeq is my object, and the $ ID is referencing to that first column. The first column, the header for the first column is ID. So all I need to do is to change this AS the character, and then also put that column in parentheses. So these change what's it called, data type from a factor data type to a character data type. So these allows us to perform a downstream analysis. I think I have some messages. If it's been an error message. All right, we'll fix that. I'll go back to, for those having errors, just want to run through it, and then we'll go back to fixing the issues. So once you have the first column now converted into the correct data type, you're good to go. So the first thing you want to do is to normalize, in this case, standardize our data set. So we don't want to make up for outliers, because sometimes this experimental data comes with too big of a number. For example, let me just let's look at the data set. You can just visualize that and see how this looks like. So these, okay, well, these are very close. You have the 436, you have 193, not too, and then you have 12. Imagine this one gene has 1,000 value, you have 20, and then you have 12. That's kind of like big difference. So that's why we do standardization or normalization. So in this case, what we're doing is to set the mean value for each observation to 0, and then the standard deviation to 1. So that makes all the numbers look kind of, like a normal, what's it called, graph. So you make them come to the center. So they're very close together. So that this one liner allows you to normalize or standardize your data, the entire data set. So said I'll be able to go through each. So you have to like capture this whole script together because they all function together to normalize your data set. So you have this, you can run that. And then now my data set is normalized. So what we can do again is to um, see what it looks like after this. So I'm just going to type RefSeq. Sorry, no RefSeq now because I've changed. What's it called? Initially, the object I'm working with is RefSeq, but now that I've standardized the data set, it's now called data underscore ST. So now if you look at now this is the new data set, and just to visualize what it looks like, if you look at the numbers, they are not like, we're not seeing 1,000, we're not seeing 12. So they're kind of very close from 0 to 1 to -1. So it falls within that range. All right, so that's very important when working with DEGs. All right, so it's been done. And then now the next thing is to create data metrics. So the data metrics, I said, allows for quantification of the gene. So now I want to represent the gene by the p-values, by the FDR, by their fold count, and all that. So this is the one-liner code for that. I know this is something anyone can use. Once you have all this, this is a pipeline. You can just copy and paste if you're working on RNASeq. You know, most people do. You might be interested in this. So this is the one-liner to create the data metrics. All right, so it works fine. So now I have my data metrics. And now this also it's being put in a different object. So the object for that is so now I've created what's it called, data metrics, and then the object for that is the data underscore st underscore sub metrics. So that's a new data for that. So that would have the p-values for each observations, the p-values, the FDR for each observation, and if the log for the fold counts for each observation. So one thing I just want to do real quick is to start plotting the first graph. So first thing is to plot two clusters. So we're interested in two clusters. By two clusters, we want down-regulated genes and the up-regulated genes. So this is that for plotting that. And then let me just, once you run this line, yeah. So I have as a new pop-up window. I'm not sure if you guys can see it, but it just pop up. So these from my ooh. Okay, so from the first graph, set plot two clusters. So by two clusters, we have the up regulated, which is the red one, and the down regulated, which is the blue one. But in case anyone is kind of interested in how does it do that? So remember by the data metrics, I was able to quantify the each observation. So the numbers are now represented by p-values and the FDR. So most times for differentially expressed genes, we use the FDR by default. So you might want to set a threshold. So which means like once you have those numbers, you want to set a threshold like, okay, so I want genes that are like within most times when it comes to the FDR rate, we're looking at 0.05 or 0.1, but in this case, I'm using 0.1. Any gene that has values outside of that are differentially expressed. So that's if you look at this function, the plot clusters. So I have the data set, and then I include the data metrics, which is this, and then I include the threshold value, which is 0.1, and the number of clusters that I want is 2. If you want more than 2 clusters, you can indicate that here, but you have to also change the number of colors that you want. And then this command, verbose, helps to print the cluster into separate files. So that also is stored in your temporary folder on your computer. So in case you want to access those files later. And then once you have all that into this object, which is known as the rect, you can then easily plot the rect C1 to D7. Remember, it's Control 1 all the way to D7, and then to clusters. So that's what that code is doing. And then if you look at the cluster 1 genes, the number is 11, and cluster 2 genes, the number is 2. For cluster 1, we might not be able to see the individual genes, but if you look at cluster 2, these kind of map, what's it called? The individual, each line represent each observation, and each observation represent a gene. So let's say, for example, you're looking at a different genes that are expressed in the case of a kidney cancer. So in this case, for cluster 2 genes, all you know is you have this one line, and if you look at how it started, even though it goes down and then up and then all the way down, but in the treatment group, you can see that the value is lower compared to in the control. And this is different from cluster 1 gene, because if you look at the value of each individual genes, it started from around negative minus 0.5, and then it goes up to almost 1.5. So that's an increase, right? So these are up-regulated genes as you move from the control sample to the treatment sample. And cluster 2 gene represent the down-regulated genes as you move from the control sample to the treatment samples. So I'm not sure if you guys, I'm going to close this so I can move on to the next one. And then one good thing, you might not be able to see this, is so we have this list of genes that are differentially expressed, but you don't know what genes are differentially expressed. You can actually print out the list of those genes and store them in your temporary directory on your computer. So these commands will allow you to do that. So you have the cluster 1, you have the cluster 2. Cluster 1 will print out the upregulated genes, store them in a file, and cluster 2 will print out the downregulated genes and put them in a file. So once you're done with the analysis, you can actually go into those files to see the names of those genes that are differentially expressed. And this is also important if you have to publish those beautiful figures. You may also indicate the names of the genes that are differentially expressed. All right, so I ran those 2 lines. So I have the list of these differentially expressed genes. And now the next is to plot a scatter plot matrix with the DEG clusters. So first I want to start with cluster 1. So these all command allows you to, let me see, allows you to plot cluster 1. Now the next one will be for cluster 2. So remember, the cluster 1 is represented by red and cluster 2 is blue. So let me just start with this and run the first cluster. So it's 6%, 80%, and 100%. All right. So these shows you, oh, okay. So this shows you a scatter plot of the upregulated genes in red. You can see, so each dot represents a gene, and of course the red line, which is the regression line. So most of the genes are supposed to be very close to that line. So look at this, what's it called? This case is we're looking at C1.1 against C1.2. So you can see how those genes are kind of distributed within this. So these are just a scatter plot representing different, but we could see at least here, because it's still within the control, you can see that they're still very close to each other. But as we move from control to treatment, so let's look at this. This is C1.3 against D7.1. We could see that it begin to move on one side of the regression line. And then the same thing applies to here. And look at these, look at the red genes, look at the red genes, look at the red genes, and look at the red genes. So as you move from control or from control to experiment, you will see that these genes are kind of away from that line. But within the control, you can see that those genes are very close to the line. So that's kind of notes. Another way to visualize your DEG, another way to also explain what you've discovered in your analysis. All right, so that's for cluster one. Just going to do the same for cluster two. Plot the scatter plot. Let me see. And that's X, 50, 60, 70, 100. All right, so that should come up as well. And then we could see the same for the blue as well. So these are the blues. You can see they're just two. So in this case, the two is so hard to tell, but I think that's why bioinformatics, it's very important, and I'll make it easier for you to analyze all this easily. So that's that for that. Without wasting more time, let me just take you guys to some more interesting plots that you can use to visualize your findings. So this one is the volcano plot, and then I'm going to plot for the for cluster one and then cluster two, and then call it a day. So so this is cluster one. So cluster one, take into consideration the p-value against the log of the fold count, and you can see these are the differentially expressed genes. They're kind of away from usually these genes very close to this zero are not, they're the same across control and experiment. But if you look at this, they're farther away, and this shows that they're differentially expressed. So that way to you can easily see that on here. Without wasting much time, let me just show you that for downregulated. Since it was just two, all right, this is just two. So you could see the blue color. I know the blue against the blue background is not really visible. You can see these two genes. You can see they're very close to one another, and they're kind of farther away from the midline, which is this zero, compared to the other one. So these are the different figures. Few of the figures you can draw using using the bigPint to visualize differentially expressed genes. You can, there's so many others that you can do, but just for the sake of time and without boring you guys. So you can see the volcano plot, you can see the scatter plot, the scatter matrix for the differentially expressed genes. All right, so that's it for the bigPint data set, bigPint our package. If anybody have any questions, oh yes, people do have questions. So for Ms. Rashid, right? Okay, Rashid, and let's see, and Kwesi, right? I'm not sure. Houston. So you said you ran into errors when you're doing this first step. I guess it's how to install and library the bigPint package. So the first thing, yes. So the first thing you want to do is make sure you run all these three lines together. Make sure they're like together like this. So you have to run these three lines because this is just one liner, but because it can't extend beyond this level, so we have to like break it into three lines. But when you're running this, make sure you highlight these three lines, and then just run the line, and that should install the bigPint. And once you're done installing the bigPint, you see a prompt window, not really a window, right on the console, asks you to update all of your packages. So this is where the A comes in handy. So the A is just a short for all. So once you run the all, it tells R to update all of the packages. So that's what this is for. And then the same applies to Edge R as well. Once you're done installing, it's going to prompt you to update the Edge R package. So you might want to choose the A as well. I hope that fixed. I hope that fixed your error. You might want to try that again and see if it fixed that. If it doesn't, I guess we'll probably have to settle that in the breakout rooms. All right. So does anyone have any question regarding this package?
Okay, so I just had another question.
Yes.
So oh, so over here it says that, like, the update prompt does not appear. It says packages not installed when version same as current. Use force equals true to reinstall bigPint. And so, like, the prompt where it appears, like, do you want to update all of your packages, some or none? Like, that prompt doesn't appear. So so as a result, when I press A, or have the A in my command, it doesn't register.
Oh, okay. So you can try the none. I guess maybe you're using a newer version of R. Maybe that's why.
Okay, okay.
All right.
All right, thank you.
You're welcome.
And so the error basically came when I typed in RefSeq with read.table,
Yeah.
and
Okay.
so that was the only part where, like, I encountered errors.
Oh,
So
this part. All right. Okay. So the first thing you want to do, Rashid, is in the beginning, I did give out this command set WD, which is set to working directory. So the folder where you have all these files stored in, you should be able to provide a file path for that folder, like I did. In my own case, this is the folder where I have the subset.txt file. Do you have the subset.txt file?
oh, I think that's what the problem is. I don't have the file, and so, like, I think
Oh,
that's.
check
Oh,
the
okay, thank you
chat.
so much.
Check the chat.
Okay, yes. So I did check chats just now, and so basically I'm downloading the file from here.
Okay, okay. Once you have the file, I think it should work.
Okay,
Mhm.
it makes much more sense now. Thank you.
You're welcome. And thanks for the question.
So Adebiyi, do you finish?
Yes.
Okay.